Okay, so now in this video lecture we will going to discuss about another kind of library, that is cDNA library. Okay, so we have already discussed about the concept of genomic DNA library, right? So I would like to recommend you, if you have not watched my genomic DNA library video, or in case you have not any knowledge about what is genomic DNA library, then I will like to recommend you to go into my YouTube channel and watch the video about the genomic DNA library, so that you can get some knowledge about how a genomic library is constructed. Okay, so now we will going to discuss about cDNA library. Okay, so again in this video I will going to ask you the same question, that is, what comes in your mind when I will tell you about library. So library means a large room consists of variety of books, right? So now, under such circumstances, let's say you are in a library, and in that particular library there is a wide portion which do not containing any kind of books. Okay, instead of, it contains only the cover of the books or any kind of things, that is not we can call as a entire book. Okay, because it can be possible, right? In some cases there can be bunch of files or bunch of project files present without any important documents. Okay, so is those stuffs are needed for your library? Is you, as a librarian, you would like to have a large portion which only contains some covers or some debris of the book? No. Similar case is applicable for genomic DNA library. Okay, because I hope you know that in whenever we are discussing about eukaryotic cell, so eukaryotic cell genome contain two kinds of gene. One is exons, that I have mentioned here by filling the boxes, and another kind of things are intron. So there are various kind of exon portion and various kind of intron portions available. Okay, so now let me introduce that these intron portions are those kind of debris that I have mentioned. Okay, so that means these intron are non-coding genes, for example some regulatory gene stuff. Okay, so now under such circumstances, do we need those kind of introns? Because we have to consider separate kind of hosts for them, right? So it will be completely a wastage, right? So under such circumstances, it was become very important to remove those kind of introns. Okay, so now what can we do? So there are two kind of processes. So one thing we can do, that we will cut each of the intron with the help of restriction endonuclease. Okay, and then we will arrange each of the exons. So here we have 5 exons, so we will arrange them, right? So although it seems quite easy, but it is not easy, because just try to think about, this is, suppose your one exon. Do you know what in is its 5 prime end and what is its 3 prime end? No, it is not possible to know. So under such circumstances, you have to use various combinations, right? So as here 5 exons present, so the number of possible combination will be 5 factorial. That means 5 into 4 into 3 into 2 into 1. That means in total we have to analyze 120 combinations. Okay, and that is not easy, right? So this process was not adopted. Now under such circumstances we have to develop another procedure. So what should we do? So whenever we are in serious problem, God saves us from that kind of serious problems. So here God again help us and shows us the way out. Okay, so he suggested to analyze the mRNA very carefully. Okay, and he also suggests us that use a enzyme named reverse transcriptase. Okay, so let me told you that reverse transcriptase is a RNA dependent DNA polymerase. What is the meaning of the term RNA dependent DNA polymerase? That means suppose that this black line is a mRNA or any kind of RNA molecule. Okay, under such circumstances, if we apply reverse transcriptase, then it will generate a DNA molecule which will be exactly complementary to the mRNA molecule. Okay, why? Because this reverse transcriptase uses this mRNA molecule as a template and creates a single stranded DNA molecule. Okay, now we are end up with a DNA hybrid which contains a cDNA. Now why the name cDNA? I hope you can understand, because it is the complementary of the mRNA, right? So we are end up with a complementary DNA molecule. So now if we will then fragment this complementary DNA molecule into number of small fragments and clone each of them, so what we will get? We will get a library, right? And this library will consist of a set of recombinant clones which will contain what? Which will contain the entire DNA sequence that have been transcribed into that mRNA molecule, right? So that means we can construct a DNA library which will not contain any kind of introns. Okay, so in this video we will going to discuss about how a cDNA can be constructed. Okay, so the first hurdle is we have to first isolate the RNA mixture from the entire cell. Okay, so we have isolated RNA. Now let me told you that RNA is not only of mRNA type. Okay, there are also various kind of RNA present. For example, we have tRNA, that is our transfer RNA, and play a important role in protein synthesis, and in ribosomal RNA, which also helps in protein synthesis, right? So now what we need, because remember only mRNA is responsible for the protein production, okay, tRNA and rRNA not. So we need only mRNAs. So our first goal should be to isolate the mRNA from those tRNA and rRNA molecule, and this is going to be a very hard task, because the size and shape of the mRNA, tRNA, rRNA molecule are quite same, despite that tRNA have to overlap structure, but still. Okay, so under such circumstances we cannot use any kind of agarose gel electrophoresis techniques, right, or any kind of bioinstrumentation technique we have. So this again become very difficult, okay, almost impossible situation. So under such circumstances God again help us. He again suggested us that please look at the 3' end of the mRNA. Okay, so God suggested us to please analyze carefully the 3' end of the mRNA. So after taking the suggestion from God, we have find out that mRNA contains a poly A tail at its 3' end. Okay, so scientists decided to use the God's suggestion to extract mRNA from that complex mixture. Okay, so the idea is they have developed a chromatographic technique known as oligo-DT cellulose chromatography. So what is the idea? So here we are using simply oligo-DT beads as a ligand. Okay, that means what? It is simply a affinity chromatography. We have prepared a stationary phase which contains oligo-DT beads. Okay, so now under such circumstances, just try to think about if we will add the entire RNA mixture in that column. Okay, so as tRNA and rRNA are devoid of poly A tail, so only mRNA will be attached here. Okay, and become entrapped in that solid space. Okay, so we can say that all the rRNA and tRNA present in the mixture will be removed, right? And then we will elute them. That means we will elute the mRNA molecule. Okay, so that means we have successfully isolated the mRNA molecule from a complex cell mixture. First step is the total RNA sample separation. Then from that we will separating the mRNA with the help of oligo-DT cellulose chromatography. Now we have mRNA molecule. Okay, so now what we will do, see very carefully. So we will add a primer at the 3' end of the mRNA molecule. Now try to understand it very carefully that as we know that the 3' end of the mRNA contains poly A tail, so we can easily prepare a primer which will contain a oligo-DT nucleotides. That means it will contain a poly T primer, right? That means a short stretch of oligonucleotide which will only consist of T. Okay, now why we are providing the primer? Because remember, any kind of polymerase cannot initiate the DNA synthesis. Okay, it only have the ability to perform the extension if it somehow gets a 3' OH group from anywhere. Okay, so in the normal DNA synthesis, that is the our replication process, natural replication process, the 3' OH group is provided by the primase, right? So now as here we are doing it totally in vitro, so we have to provide the 3' OH group. That is why we are constructing the primer first. Okay, and here our primer will only contain the T nucleotides. Okay, so now under such circumstances we will add reverse transcriptase, and I have told you that this reverse transcriptase is a RNA dependent DNA polymerase. That means what? The reverse transcriptase will use this mRNA molecule as a template and construct a cDNA molecule. Okay, so now what we have? We have a RNA DNA hybrid. Now what we will do, try to understand very carefully. So now we have RNA DNA hybrid. So do we need RNA? No. So first goal of us should be to remove the RNA, right? So we will apply alkali treatment, or we can use also RNase H. I have told about RNase H in the enzymes in recombinant DNA technology video, where we have discussed that we can remove RNA from a RNA DNA hybrid with the help of that enzyme. Okay, not only that, our retroviruses, for example HIV viruses, also uses RNase H. Okay, so what RNase H will do? RNase H will remove that poly A tail containing mRNA. So now we have only single stranded cDNA molecule. Okay, now let's try to remember about another enzyme that we have also discussed that can add nucleotide molecule template independently at the 3' end. So this enzyme, the name of that enzyme is our terminal deoxynucleotide transferase. We have also discussed about TDT in enzymes in recombinant DNA technology video. So now we are having a cDNA molecule which contains a polyG at its 3' end, and also we have poly T nucleotides at the 5' end of the cDNA. Okay, so now again we know. So now can we use that cDNA molecule for cloning? No, because it is a single stranded cDNA molecule. So now our goal should be to convert it into a double stranded cDNA molecule, right? So again, as we know what is present at the 3' end of the single stranded cDNA molecule, can we construct a primer? Yes, we can construct, and that primer will only contain the poly C nucleotides, right? So now on the basis of that primer we can use normal DNA polymerase. That DNA polymerase will extend the primer and leads to the formation of a double stranded cDNA molecule. Okay. Okay. So now we are having this type of situation. Okay. So now let me discuss that. Suppose we don't know about what is the sequence of that DNA molecule. Okay, so under such circumstances the protection of that DNA should be necessary. Okay, because remember here we are transferring that DNA inside a host cell. Okay, and we all know that bacteria have restriction modification system, and which consists of two parts. One is restriction, another one is modification. Okay, and this restriction part is none other than our restriction endonuclease. And now if by chance that gene of interest contain a recognition sequence for the restriction endonuclease, what will happen? It will simply chew it up, right? And as a result our gene of interest will be lost, right? So we have to provide extra attention on them, right? Not only it will be chewed it up by our host, but it can also be chewed it up, majorly can be chewed it up with the help of the restriction endonuclease we will use for the isolation of our gene of interest whenever we are requiring after the construction of the DNA library. Not only that, also for whenever we will use, or also when we will going to use our restriction endonuclease treatment step for the ligation into the vector. Okay, so that is why the protection of that gene of interest is necessary. So under such circumstances we will use various kind of methylase enzyme that have the ability to methylate it. Okay, because remember restriction endonuclease do not cause any kind of chewing activity towards any gene of interest which is fully methylated or heavy methylated. Okay, so methylation of both the strand is necessary. So we have methylated with the help of methylase enzyme. We have two kind of methylase enzyme, DAM and DCM methylase enzyme. Okay, so now we are ready for insertion, right? Now let's look at the 5' or 3' end. Is they are suitable to be a restriction cut site for any kind of restriction endonuclease? Do you have any kind of restriction endonuclease that contains sequences just like that? No, obviously not. So here we have to use the linker. Okay, so linkers are what? Linkers are short nucleotide sequence which contain restriction recognition site for a number of different restriction endonucleases. Okay, so here we will going to use EcoR1 linker. Okay, so that means what? EcoR1 linker is nothing but a short nucleotide sequence which will contain the restriction or restriction site or recognition site for EcoR1 enzyme. Okay, so let me discuss that the recognition site for EcoR1 is that kind of palindromic sequences. So we will going to use this short stretch of nucleotide sequence as a linker. Okay, and that will be known as EcoR1 linker. Okay, so we have used those kind of linkers as you can see in this picture. Okay, so now we can easily cleave it with EcoR1. Okay, now just try to think about this. If we do not methylate them before that cleavage step, and by chancely if the gene of interest will contain something like this GAATTC CTTAAG, then the EcoR1 will not only cleave the linkers but it will also shoot it up the gene of interest. As a result, the gene of interest will become lost, right? So that is why we have methylated. So now we have our staggered in, right, and we will ligate them into a lambda arms and package in vitro, and we have infected E. coli cells with the help of those kind of vector containing gene of interest. Okay, now by plating those kind of E. coli in a LB agar medium containing appropriate antibiotic, let's say ampicillin, we can construct the cDNA library, okay, which will consist of a set of different recombinant clones. Okay, so each of the clones will be known as cDNA clones, and the entire set of those cDNA clones are collectively known as cDNA library. Okay, so that is the concept of cDNA library. That is the concept how we can construct a library of gene which will not contain any kind of introns or non-coding genes. Okay, so now if you have understand the entire concept of cDNA library, then I would like to discuss with you about a important and interesting difference between genomic DNA library and cDNA library. Okay, so first thing that I want to discuss, that what do you think, is a size of cDNA library will is a size of cDNA will be equal to the genomic DNA? Is it? No, because cDNA will only contain the exons. On the other hand, our genomic DNA library that I have mentioning here with a red color will contain mixture of genomic exon and introns, right? So now this thing is only applicable for eukaryotes. Okay, because remember it again that eukaryotes will contain introns as well as exons in their genomic DNA. Okay, but cDNA lacks it, right? That means cDNA lacks introns. On the other hand, if someone asks you that whether the genomic DNA size and the cDNA size for a prokaryote will be same or not, so in that case your answer will be yes, the size will be same, because remember it very carefully that genomic DNA for prokaryotes only contains exons. Okay, they lacks introns. Okay, so that is the concept. Another important difference that I want to mention, that for genomic DNA library construction of an entire organism, you have to clone the entire DNA of a single cell only. Okay, because the entire DNA composition of a cell of a eukaryotes are more or less same with the every other cell present in the entire body. Okay, so the genomic DNA library constructed on the basis of a particular cell will be enough for the genomic DNA library of the entire organism. Okay, but this is not applicable for cDNA library. Let me discuss how. Because remember, the cell which is present in the liver tissue and the cell which is present in the kidney tissue, both will transcribe various kind of mRNAs, right? And those mRNAs will produce protein, and depending on those protein, the function of the kidney and the function of the liver are different, right? So can I expect that the cell which is present in the liver will transcribe mRNA molecules which are exactly the same that are transcribed by a cell present in kidney? No, it is naturally impossible, because if the cell present in kidney will transcribe a set of mRNA molecule which is exactly the same with the cells which is present in the liver, then the function of the liver and the function of the kidney will be same. But is it really possible? No. That means what? That means a cDNA library that you have generated only reflects the entire DNA sequence that are transcribing the entire mRNA molecule of that particular cell only. Okay, that means what? That means that particular cDNA library that you have constructed is only specific for that particular cell that you have used to construct the cDNA library. Okay, on the other hand, if one genomic DNA library will represent the entire or genome of an organism. Okay, so that is the idea you have to remember. So let me discuss it diagrammatically, then you can better understand. Suppose there is a cell A which is from a liver. Okay, on the other hand, there is a cell B which is from kidney. Okay, so now inside the cell there are various mRNAs transcribing. Similarly, inside the B there are also various mRNAs transcribing. Okay, so now the A will produce protein which will play important role in liver. On the other hand, B will produce protein which will play important role in kidney. Okay, so under such circumstances, I can expect that the mRNA molecule produced by the gene A, mRNA molecule produced by the cell A should be different from the mRNA molecules produced by the cell B, and as a result, can I expect that the mRNA molecule that coming from a particular DNA should be different from the mRNA molecule that is coming from a particular DNA in cell B, and as a result we can conclude that the gene A which is present inside the cell and which are responsible for the transcription of the mRNA molecule inside the cell A should be different from the DNA molecule present in the cell B which are responsible for the transcription of that particular mRNA, right? So under such circumstances, we can conclude that the cDNA library we have constructed on the basis of gene A should be differ from the cDNA library we have constructed on the basis of gene B. Okay, so that was the entire concept that I have written here. So let us read the concept again, then it will be super clear to you. So there are some differences between genomic DNA clone and cDNA clone. So genomic DNA clone represent a random sample of the entire DNA sequence in an organism, and with very rare exception are the same regardless of the cell type used to prepare them. That means with the help of a single cell or one type of only a one kind of cell, we can construct genomic DNA library which will represent the entire organism. On the other hand, by contrast, cDNA clone will only contain those genes which are transcribing the mRNA, right? So because the cell of different tissues produces distinct set of mRNA, so it is obvious that distinct cDNA library is obtained for each type of cell used to prepare the library. Okay, so that is the concept, right? So that is all for the construction of the cDNA and the cDNA library. So I hope this video will be helpful to you to understanding the concept of cDNA library, and thank you for listening to this class.